DNA and its importance in living organisms. Deoxyribonucleic acid, DNA, is the fundamental molecule that carries the genetic instructions essential for the formation and development of living organisms. This molecule serves as the biological code that determines inherited traits such as eye color, hair type, and height. DNA plays a crucial role in vital processes like growth, reproduction, and cell repair, making it the cornerstone of life on Earth. DNA is composed of small units called nucleotides, arranged in a long chain that forms a twisted ladder-like structure, known as the double helix. Understanding DNA provides scientists with powerful tools to uncover the origins of genetic diseases, develop innovative treatments, and even enhance agricultural crops through genetic engineering. The significance of DNA in living organisms. Transmission of genetic traits. DNA transfers genetic information from one generation to the next, ensuring the continuity of unique characteristics in organisms. Regulation of biological processes. It contains the instructions for producing proteins, which are essential molecules responsible for various cellular functions. Contribution to biodiversity. Genetic mutations in DNA contribute to genetic diversity, enabling organisms to adapt to different environments. Scientific research and medical applications. DNA is utilized in early disease diagnosis, vaccine development, and organ transplantation using genetic engineering techniques.
Structure of DNA. DNA is made up of basic units called nucleotides, and each nucleotide consists of three main components. Phosphate group connects to the sugar molecule and forms the backbone of the DNA strand. Pentose sugar, a sugar containing 5 carbon atoms. Phosphate group, a sugar containing 5 carbon atoms. Nitrogenous base. There are 4 types: adenine, A, thymine, T, guanine, G, cytosine, C. The nitrogenous bases pair in a specific way. Adenine, A, pairs with thymine, T, via 2 hydrogen bonds. Guanine, G, pairs with cytosine, C, via 3 hydrogen bonds. DNA takes the shape of a double helix, where 2 strands of nucleotides coil around each other in a spiral. Functions of DNA: storing genetic information. DNA holds the genetic code that determines the traits of an organism, such as hair color or eye color. Transmitting hereditary traits. DNA passes genetic information from parents to offspring during reproduction. Directing protein synthesis. DNA contains instructions for making proteins through a process called translation, which occurs after the instructions are transcribed into RNA, ribonucleic acid. Repairing damaged cells. DNA helps repair mutations or damage in cells caused by external factors like radiation or toxins. Regulating biological functions. It controls the growth and development of an organism by regulating the activity of various genes. Importance of DNA in science medicine. DNA analysis helps in detecting and diagnosing genetic disorders. Agriculture. Genetic modification techniques improve crop yield and resistance. Forensics. DNA analysis is used to identify individuals in criminal investigations. Foreign medicine. Natural-born killers transform into natural-born killers. Forensics. DNA analysis is used to identify individuals in criminal investigations.
The discovery of DNA, from an unknown molecule to a cornerstone of science. Introduction. DNA, deoxyribonucleic acid, is one of the greatest scientific discoveries in human history. It is not just an ordinary molecule, it holds the key to understanding life itself. The journey of DNA discovery evolved from being an obscure molecule to becoming the foundation that connects various fields of science, such as medicine, biology, and genetics. Historical roots of DNA discovery. The journey began in the mid-19th century when Swiss scientist Friedrich Miescher discovered a new substance within the nuclei of cells, which he named nuclein. At the time, Miescher could not have imagined that this mysterious molecule would later be identified as DNA, revolutionizing science. The double helix structure DNA remained an enigma until the mid-20th century, when scientists James Watson and Francis Crick unveiled its double helix structure in 1953. They based their discovery on X-ray diffraction data collected by Rosalind Franklin. This breakthrough marked a turning point, unlocking the secrets of how genetic information is stored and transmitted across generations. The importance of DNA in Science 1. Genetics and genomics. DNA is the cornerstone of modern genetics. It has enabled scientists to decode the genetic blueprint, leading to innovations like genetic engineering and gene editing. 2. Personalized medicine. DNA plays a critical role in advancing personalized medicine. By analyzing genetic information, doctors can predict susceptibility to diseases such as cancer and cardiovascular conditions, allowing for tailored treatments. 3. Forensic science. DNA is a powerful tool in forensic investigations, enabling the accurate identification of individuals and solving criminal cases with high precision. DNA and its relationship to heredity. DNA is a molecule that carries the genetic instructions essential for the growth, development, functioning, and reproduction of all living organisms. It serves as the instruction manual that determines inherited traits such as hair color, height, and eye color. DNA is a double helix structure composed of two strands twisted around each other. These strands are made up of units called nucleotides, which consist of three components: a nitrogenous base, adenine, thymine, guanine, cytosine; a 5-carbon sugar, diastereobose; a phosphate group. The role of DNA in heredity transmission of genetic traits. DNA is passed from parents to offspring through sex cells, eggs, and sperm. Each organism inherits 2 copies of each gene, one from the mother and one from the father. Genes are specific segments of DNA that contain instructions for producing proteins. These proteins play a crucial role in determining physical traits and performing vital biological functions. Color traits inherited. Meiosis. Zoas. During the formation of sex cells, DNA undergoes a special division process, called meiosis, which reduces the chromosome number by half, ensuring that the new organism receives a complete set of genes from both parents. Mutations. Sometimes, random changes in the DNA sequence, known as mutations, occur. These mutations can affect inherited traits or lead to genetic disorders. The importance of studying DNA. Diagnosis of genetic disorders. DNA studies help identify genes linked to diseases such as cystic fibrosis or hereditary cancer. Advances in gene therapy. Genetic defects can be corrected using techniques like CRISPR to modify DNA. Ancestry and paternity testing. DNA analysis is used to determine family relationships, such as confirming parentage.
Applications of DNA in medicine and biotechnology. DNA is one of the most crucial biomolecules that has revolutionized medicine and biotechnology. Thanks to advancements in genetic science and DNA analysis technologies, it has become possible to utilize DNA in innovative ways to improve health and develop biotechnological solutions. In terms of scientific development, DNA is one of the most crucial biomolecules that has revolutionized medicine and biotechnology. First, applications of DNA in medicine. 1. Genetic diagnosis. DNA analysis is used to detect genetic disorders, such as cystic fibrosis and sickle cell anemia. It helps identify genetic mutations that increase the risk of diseases like cancer. 2. Gene therapy. Healthy genes are introduced into a patient's cells to treat genetic disorders. It is being used to address conditions such as muscular dystrophy and certain types of cancer. 3. Personalized medicine. Personalized medicine uses DNA analysis to tailor treatments for each patient based on their genetic makeup. This approach helps determine the most effective medications while minimizing side effects. 4. Prenatal testing. DNA from the fetus is analyzed through a blood sample from the mother to detect genetic abnormalities, such as Down syndrome. Second, applications of DNA in biotechnology. 1. Genetic engineering. DNA is used to modify the genes of living organisms to produce valuable products, such as genetically modified crops that resist pests or yield more produce. 2. Production of biopharmaceuticals. DNA from microorganisms is harnessed to develop drugs like human insulin and growth factors. 3. Agricultural biotechnology. DNA technologies are applied to enhance crop production and improve resistance to harsh environmental conditions. 4. Biofuel production. Genetically modified microorganisms convert biomass into alternative energy sources, such as ethanol and biodiesel. Third, other applications of DNA. 1. Forensic science. DNA is used to identify individuals in criminal investigations and analyze forensic evidence. 2. Scientific research. DNA is a fundamental tool for studying gene functions and the evolution of living organisms. 3. Biological conservation. DNA analysis is utilized in conservation programs to monitor genetic diversity and aid in the reproduction of endangered species.
Ethical challenges associated with DNA studies. Advancements in DNA research have revolutionized fields like medicine, biology, and forensic science. However, these developments bring forth several ethical challenges that require careful consideration and management. The key ethical issues include: 1. Privacy and data confidentiality. One of the biggest concerns is safeguarding personal genetic information from unauthorized access or misuse. Risk of hacking. DNA databases are vulnerable to cyber attacks, posing a threat to individual privacy. Unlawful use. Genetic data could be exploited by insurance companies or employers to discriminate against individuals. 2. Informed consent. Collecting DNA samples requires clear and explicit consent from participants. Transparency in research. Researchers must explain the goals, risks, and potential uses of the data. Future use. Samples may be used for future studies not initially disclosed, raising concerns about the scope of consent. 3. Genetic discrimination. Genetic information can lead to discrimination based on an individual's predisposition to certain diseases. Health insurance. Insurance providers may deny coverage or increase premiums based on genetic findings. Employment. Employers might discriminate against individuals with a higher risk of developing certain health conditions. 4. Gene editing and the ethics of genetic modification. Techniques like CRISPR allow for the modification of genes to treat diseases or enhance traits. Medical ethics. Is it ethical to alter human genes for intelligence or appearance enhancements? Unintended consequences. Gene editing might result in unforeseen side effects or health problems for future generations. 5. Impact on family relationships. DNA tests can reveal sensitive information, such as non-paternity or unknown relatives. Family strain. Such discoveries may cause conflicts or change family dynamics. Adopted individuals. They might face emotional distress upon learning their biological origins unexpectedly. 6. Genetic research and racial minorities. DNA studies might be misused to justify racial or ethnic discrimination. Scientific misuse. Some studies could be exploited to claim genetic superiority or inferiority of certain groups. Research trust. Minorities often fear their genetic data might be used without consent or against their interests. 7. Clinical trials and exploitation of vulnerable populations. Genetic research might target poor or vulnerable communities for clinical trials without adequate protections. Social justice. The benefits of research should be distributed fairly, avoiding exploitation. Unanticipated risks. Participants could face side effects or unknown risks during trials.
Struggles of scientists to decipher the human genome. Over the past decades, the world has witnessed a fierce race to unlock the secrets of the human genome, a project considered the key to understanding the genetic blueprint of humanity. This race was not merely a scientific challenge, but also filled with conflicts among scientists, research institutions, and even nations. The beginning of the race to decode the genome. In the early 1990s, the Human Genome Project was launched as an ambitious international collaboration aimed at sequencing the entire human DNA. Led by governments and major universities, it was seen as a revolutionary step toward understanding genetic diseases and developing innovative treatments. Public vs. private sector competition. The competition quickly heated up between the public Human Genome Project, led by scientists Francis Collins and James Watson, and Celera Genomics, founded by Craig Venter. While the public project relied on collective efforts and traditional sequencing methods, Celera used faster but less comprehensive sequencing techniques. Celera used faster sequencing to faster sequencing techniques. Once accepted, Celera. The battle over intellectual property. One of the most contentious issues was the question of intellectual property rights. Companies like Celera sought to patent specific genetic sequences, which angered scientists who believed that genetic knowledge should belong to humanity as a whole. Scientific and economic objectives. The conflicts extended beyond technical aspects to include economic and political goals. Major nations like the United States and Japan were vying for scientific and economic dominance by controlling this vital information. In this achievements despite challenges. Despite the conflicts, the completion of the first draft of the human genome was announced in 2003, marking a historic milestone. This breakthrough revolutionized the understanding of genetic disorders such as cancer and autism, and paved the way for personalized medicine. Ethical challenges for the future. With the continuous advancement in genetic science, scientists face ethical dilemmas, including genetic manipulation and genetic privacy. Balancing innovation with the protection of individual and societal rights is now more crucial than ever.
DNA and the Future of Science, Genetic Engineering and Gene Editing, Introduction. DNA, the blueprint of life, carries the genetic code for all living organisms. Since its discovery, this small molecule has revolutionized fields like medicine, agriculture, and even industry. With the advancement of genetic engineering techniques, it is now possible to modify DNA with precision, opening the door to endless possibilities for improving human life. What is genetic engineering? Genetic engineering refers to the process of altering the genes of living organisms using advanced technologies. This involves inserting, deleting, or modifying specific genes to achieve desired traits. These techniques are used in various fields, from enhancing crop yields to treating genetic disorders. Key tools in genetic engineering: CRISPR-Cas9 technology. CRISPR is a groundbreaking tool that allows precise gene editing. It is used to correct genetic disorders such as cystic fibrosis, or even combat cancer. Recombinant DNA technology. This involves combining genes from different organisms, enabling the production of biopharmaceuticals like insulin and vaccines. Benefits of gene editing. 1. In medicine treating genetic disorders, gene editing can correct mutations responsible for diseases such as thalassemia and sickle cell anemia. Immunotherapy for cancer. Genetic modifications enhance the immune system's ability to target and destroy cancer cells. 2. In agriculture disease-resistant crops, gene editing helps produce plants that are more resistant to drought and pests, improving global food security. Enhanced nutritional value. Crops can be fortified with vitamins and minerals to address malnutrition. 3. In industry biofuel production, engineered microbes can convert waste into clean energy sources like biofuels. Innovative materials. Gene editing is used to create materials like synthetic spider silk, known for its strength and flexibility. Challenges and ethical considerations. Despite its immense benefits, genetic engineering raises numerous ethical and legal concerns. Environmental risks. Editing genes in living organisms could have unforeseen ecological consequences. Manipulating human genes sparks debates, particularly concerning non-medical uses like enhancing intelligence or appearance. Genetic privacy. There are concerns about the misuse of genetic information for commercial or discriminatory purposes. Future. What does gene editing hold for us? As genetic engineering technologies continue to evolve, they promise unprecedented advancements across various fields. Curing incurable diseases. Diseases once deemed incurable might become treatable. Extending human lifespan. Gene modifications could slow aging and enhance the quality of life. Reviving extinct species. DNA technology may enable the resurrection of extinct species, raising questions about its impact on ecosystems. How DNA changes human lives. 1. Medical advancements and gene therapy. Advances in understanding DNA have revolutionized medicine. Early diagnosis of genetic diseases. DNA can be analyzed to detect mutations responsible for diseases like cystic fibrosis or Huntington's disease. Gene therapy. This technique uses DNA to correct defective genes. For instance, treatments for genetic disorders like muscular dystrophy are being developed using technologies like CRISPR. 2. Improved agriculture and nutrition. DNA-based genetic modification has enhanced the quality of crops and livestock. Genetically modified crops, such as golden rice, which contains vitamin A to combat deficiencies in certain regions. Genetically enhanced animals. DNA editing helps improve milk and meat production. 3. Personalized medicine. By analyzing an individual's DNA, customized treatments and medications can be developed to suit their unique genetic makeup. This minimizes side effects and improves the effectiveness of therapy. 4. Forensic science and crime-solving. DNA analysis is a powerful tool in criminal investigations. It can be used to identify individuals at crime scenes or to analyze ancient human remains. 5. Tracing human origins and migration. DNA research has helped trace human ancestry and migration patterns across the globe. Services like 23andMe allow individuals to explore their ethnic and historical roots. Psychological and social impacts. Learning about one's genetic makeup can influence how people approach their health and lifestyle. However, it also raises ethical concerns, such as genetic discrimination or misuse of genetic data. Conclusion. DNA is more than a biological blueprint. It is a key to understanding human nature and improving our lives through science, medicine, and technology. As research continues, DNA will likely open new doors to solving global challenges and enhancing quality of life.